Good morning, everyone. Today I will be discussing the applications of biotechnology and how it is revolutionizing healthcare and therapy. Let's begin by examining how biotechnology is transforming these fields. Biotechnology is a science that has seen many advances recently. It has allowed us to better understand biology, develop treatments for diseases, and even produce food in a more effective and sustainable manner. Moreover, biotechnology has been applied to other areas, such as producing renewable energy and increasing the efficiency of industrial processes. Breakthroughs in this field keep on appearing, reminding us that the potential of biotechnology is immense. Biotechnology is an incredibly powerful tool, one that has enabled the development of innovative treatments and medicines. Recombinant DNA technology has made it possible for us to generate safe and more effective drugs on a large scale, and its influence on healthcare is undeniable, positively impacting the lives of countless patients. Biotech has revolutionized the way we view medical treatment and diagnosis. Cloning DNA has provided us with the capability to control and manipulate the functions of the human body. This technology has allowed for the creation of hormones like insulin and interferons. Through the use of vaccines, these hormones can be used to combat microbes and other pathogens, resulting in an accelerated and more thorough treatment of sickness and infection. Biotechnology is a rapidly growing field that is being applied to numerous industries, especially those that produce commodities such as pharmaceuticals, food, energy, chemicals, and other industrial products. It leverages microorganisms and enzymes to create novel products with desired properties, such as higher efficiency and lower costs. This, in turn, increases affordability of the commodities for the wider public. Biotechnology has had a profound impact on modern medicine, providing tools to engineer targeted therapies which can address the cause of many medical conditions. These therapies, known as recombinant therapeutics, are created by introducing genetic material from one organism into another, offering the advantage of being highly specific and avoiding unwanted immunological responses that can come from non-human sourced products. Globally, 30 recombinant therapeutics have been approved for human use, with 12 of them currently available in India. Biotechnology has been a key factor in the revolution of insulin production, a life-saving hormone for those with diabetes. In the past, insulin was obtained from the pancreas of animals that were slaughtered. Human insulin is now generated through sophisticated biotechnology techniques, but its structure is the same as the organic molecule. This progress not only reduces the cost to make insulin, but also eliminates the risk of contamination and the possibility of transmitting animal diseases. Biotechnology is a powerful and versatile tool employed in modern science and medicine to remarkable effect. A prime example of this is the production of human insulin. In 1983, American company Eli Lilly manufactured 2 DNA sequences aligned to the A and B chains of human insulin, which were then implemented into plasmids of E. coli. These chains were extracted and amalgamated, forming human insulin through the utility of disulfide bonds. This pioneering accomplishment has been of great benefit to those affected by diabetes. Biotechnology is offering progressive answers to the rising worldwide requests for improved medical treatments. One model of this is the manufacturing of insulin, which has been made more available and economically feasible through novel biotechnological techniques. This has enabled better health results and a steadier market cost for the medication. Gene therapy is an approach to treating hereditary diseases that has the potential to transform the future of healthcare. It involves carefully inserting genes into a person's cells and tissues, in order to help resolve a wide range of medical issues. This type of therapy offers hope to those living with genetic conditions, and with further research and development, it may provide unprecedented treatment options to people all around the world. Biotechnology is a rapidly expanding field with a range of exciting applications in many areas of medicine. It involves introducing a normal gene into a person or embryo, to substitute and compensate for a non-functional gene. Nanomedicine is thus revolutionizing healthcare, providing practical solutions for curing diseases and restoring health. Consequently, it is making new and innovative treatments available to a wider range of people. Molecular diagnostics is an advanced technology that offers precise diagnosis of diseases in the early stages. It works by analyzing a patient's tissue or bodily fluids to determine the underlying cause of the disease. A molecular diagnosis report provides a comprehensive description of the causative microorganism, its origin, effects, and current location, making it possible to provide optimal care. In contrast, traditional methods such as serum and urine analysis do not provide the same level of information for early diagnosis. Exploring how biotechnology and its applications have enabled us to diagnose diseases earlier, intervening before symptoms emerge, recombinant DNA technology, polymerase chain reaction and enzyme-linked immunosorbent assay are three techniques that have advanced the field significantly. These techniques allow us to detect the presence of a pathogen before a disease symptom appears, meaning we are able to intervene before the concentration of the pathogen is too high in the body. This has revolutionized disease diagnosis and saved many lives. Polymerase chain reaction, commonly referred to as PCR, is a laboratory tool that has drastically changed multiple scientific fields. It is often used when examining genetic material. PCR has many applications, such as detecting HIV in patients with possible AIDS, analyzing genetic mutations in cancer patients, and detecting many different genetic conditions. This cutting-edge technique has increased the chance for earlier diagnosis and became an integral component of contemporary medical treatments. Biotechnology has been employed to create enzyme-linked immunosorbent assay, ELISA, a type of immunoassay. ELISA enables the detection and measurement of certain antigens, proteins, hormones, or antibodies in samples. This technique works according to the principle of antigen-antibody interaction. It is commonly used to identify infections by determining the presence of antibodies against a pathogen in the sample, as well as to measure the concentration of antigens. ELISA has been proven to be an effective tool to detect and quantify antigens, such as proteins or hormones. Biotechnology has many applications, one of the most prominent of which is its use in forensic science. DNA fingerprinting is a widely used approach to determine an individual's genetic identity and compare it to the DNA of other individuals, which allows for the identification of perpetrators of crimes or the resolution of parentage claims. This technology has played a great role in the development of forensic science and will undoubtedly continue to be beneficial for many years. Biotechnology is employed to modify organisms or components thereof to create new products and processes. Notably, biotechnology is manifested in the manufacture of insulin, a hormone tasked with regulating the blood sugar levels. This slide addresses the number of chains in a single insulin molecule. The answer is 4. These 4 chains unite to form the insulin molecule, a complex and unique process only achievable through biotechnology. Gene therapy is one of the most fascinating areas of biotechnology usage. It is a form of treatment permitting us to confront numerous medical complications, including age-related illnesses, genetically inherited ailments, and neurological disorders, through furnishing rectification methods involving the alteration or replacement of flawed genes. This technology affords us the capability to specifically pinpoint and modify impaired tissues or cells, giving a chance to elevate the general well-being of people. ELISA, enzyme-linked immunosorbent assay, is a biochemical technique that is commonly employed to quantify and detect specific proteins in a sample. It relies on the antigen-antibody reaction principle, wherein an antigen from the sample binds to a particular antibody in the ELISA, accompanied by a specific enzyme that amplifies the signal. To put it simply, ELISA is capable of detecting the existence of a certain antigen or antibody in a sample by specifically tying to the relevant antigen or antibody. Biotechnology is an incredibly powerful tool with a great impact on medical science. It allows us to accurately identify genetic disorders through electrophoresis, chromatography, centrifugation, and polymerase chain reaction, PCR. These techniques give us deeper knowledge of the human genome and any related disorders which may be caused by genetics or the environment. Biotechnology has enabled modification of the genetic material of plants for enhancing their yield, nutrition, disease resistance, and adaptability to environmental stress. This practice of gene transfer from one plant species to another has allowed generation of disease-resistant plants and improved nutritional value. Furthermore, transgenic plants allow efficient and cost-effective production of desirable proteins and drugs. Transgenic plants are created by artificially inserting genes that produce desirable traits into the plant's DNA. This technique is used to enhance crop yields, make crops less susceptible to diseases and modify food products. Additionally, transgenic plants can be used to make pharmaceuticals, improve industrial processes and enhance the environment. Biotechnology has enabled the transfer of genetic material from one organism to another, which has enabled the production of more targeted, productive and disease-resistant plant varieties. The T. plasmid of Agrobacterium tumefaciens is a successful vector for introducing cloned genes in plants, thus making the production of transgenic plants possible. Biotechnology has completely transformed the way crop production is approached and the way humans interact with plants. Through transgenic plants, it has become possible to cultivate crops precisely designed to cater to the needs of agriculture and human life. An example of this is transgenic tobacco. It has made it feasible to cultivate crops with a higher resistance to pests and environmental stress compared to before. This technology has given us the opportunity to hone in on specific aspects of plants to gain more understanding of the possibilities and applications of biotechnology. Biotechnology has allowed us to apply science and technology to create transgenic crop plants with improved resistance to diseases and pests. This has been attained through the recognition and isolation of genes related to certain desired traits. For instance, transgenic papaya was developed with increased immunity to papaya ring spot virus, bringing about improved productivity and enhanced fruit quality. Biotechnology is an ever-advancing field with many applications in recent times. A prime instance of this is Bt cotton, a genetically altered cotton plant that is sheltered from certain insect pests. This development has helped farmers to lessen crop losses and improve their overall yield, due to which their income has increased. The picture provided in this context shows the efficiency of Bt cotton in combating pests and elevating yields. Biotechnology is the science of modifying living organisms genetically to create new substances with certain properties. An example is a transgenic tomato plant that is resistant to bacteria like Pseudomonas. Through biotechnological methods, we can engineer plants to be more tolerant to certain illnesses and parasites, offering a secure and successful method to produce better quality food. Biotechnology is enabling significant progress in the agricultural sector, such as the development of transgenic potato plants' defense against the fungus Phytophthora. Through current technology, researchers can incorporate genes sourced from various organisms into the potato plant's gene pattern to provide it with improved immunity. This could potentially revolutionize the production and gathering of crops, making it both more effective and more economical. Biotechnology is an increasingly thriving field, especially in the area of food processing technology. One example of its application is the Flavor Saver transgenic tomato, which is specially designed to be more resilient to bruising. This provides a longer shelf life compared to normal tomatoes, as well as making it easier to safely store and transport them. This type of technology has the potential to revolutionize the availability and processing of food, making it more widely accessible. Biotechnology has enabled the production of transgenic plants with enhanced nutritional benefits, one example being transgenic golden rice from Taipei, which is rich in vitamin A. This offers not only improved nutrition, but it also reduces the risk of vitamin A deficiency-related blindness. Biotechnology has had a profound impact on the agriculture industry, providing us with the ability to develop plants with the desired characteristics. Transgenic plants are especially useful for hybrid seed creation. A prime example of this is with Brassica napus, where male sterile varieties can be produced, avoiding the need for manual emasculation and cutting down on the cost of producing hybrid seeds. This successful implementation of biotechnology highlights its capabilities for boosting a range of industries. Biotechnology is a highly effective tool with the capacity to tackle a lot of modern agricultural issues. One of its applications is making transgenic plants, which have been altered to be resistant to adverse conditions caused by various environmental elements such as chemicals, cold, drought, salt and heat. This technique was employed to a type of basmati rice, generating a strain which is resistant to both biotic and abiotic stresses. Not only does this elevate agricultural yield capability, but also improves the reliability of our food supply. Biotechnology has enabled farmers to benefit from possibilities that weren't available before, such as the Roundup-ready soybean, a plant engineered to be resistant to the Roundup herbicide. This makes it simpler for farmers to control weeds around soybean plants, thus leading to higher yields with less effort. An image illustrates the impact of this technology, resulting in increased food production. Biotechnology has been a great asset for industries in terms of manufacturing essential biochemicals. Transgenic plants have enabled scientists to encode genes for insulin, interferon, human growth hormones and antibodies. These biochemicals produced from plants have been proven to be of a same or higher standard than the ones made in bacteria, offering a remarkable source of resources for the industry. Molecular farming is a revolutionary approach to utilizing plants for large-scale production of goods such as specialized medicines, chemicals and antibodies. Its potential to revolutionize the farming and biochemical industries is becoming increasingly apparent. Biotechnology is further expanding its applications and possibilities. Biotechnology is an ever-evolving science with uses that span many industries. One of its most prominent techniques is gene transfer, which involves altering and exchanging genetic information to produce plants with specific traits. This can be achieved through a variety of techniques, such as tissue culture, transgenic plants, genetic recombination, and protoplast technology. These methods are likely to remain integral to biotechnology, providing significant advances in the future. Biotechnology is a field that is evolving at a fast pace and allows us to explore science's boundaries. It is used for a range of purposes, for example to modify organisms like plants for commercial and medical benefits. One application is the creation of transgenic plants, which are plants with genetic modifications. Some of the commonly employed vectors for this are PBR322 plasmid, COL-E1 plasmid, bacteriophage, and T plasmid. These instruments enable progress in biotechnology which would not be achievable without them. Biotechnology is a powerful tool for producing commercial products. It can be applied to a variety of methods such as biofarming, molecular farming, tissue culture, and biotechnology to enable the production of these products. By studying and manipulating these processes, a variety of commercial products can be generated that meet the needs of the people. Thank you for your attention.